about T and B cells. So the first one we'll go over is B cells. So after formation and maturation in the bone marrow, the B cells move to the lymphatic system to circulate throughout the body. In the lymphatic system, B cells encounter an antigen, which then starts the maturation process for the B cell. When the B cell encounters an antigen that fits or matches its membrane-bound antibody, it quickly divides in order to become either a memory B cell or an effector B cell, which is also called a plasma cell. Antibodies can bind to antigens directly. The antigen must effectively bind with a B cell's membrane-bound antibody in order to set off differentiation, or the process of becoming one of the new forms of a B cell. B cell receptors also play an important role in signaling pathways. After the antigen is internalized and processed, the B cell can initiate signaling pathways. This helps to communicate with other cells of the immune system. The second thing we'll talk about is T cells. So once they form in the bone marrow, T cells migrate to the thymus, where to mature and become T cells. While in the thymus, the developing T cells start to express T cell receptors and other receptors. Unlike antibodies, which can bind to antigens directly, T cells can only recognize antigens that are bound to certain receptor molecules. T cell receptors have to undergo a process called rearrangement, causing the nearly limitless recombination of a gene that expresses T cell receptors. The process of rearrangement allows for a lot of binding diversity. The diversity could potentially lead to accidental attacks against cells and molecules because some rearrangement configurations can accidentally mimic a person's self molecules and proteins. Mature T cells should recognize only foreign antigens combined with self MHC molecules in order to mount appropriate immune response.